by the sea,
by the beautiful sea.
you and me,
You and me,
oh how happy we'll be.
when each wave comes a-rolling in,
We will duck or swim.
and we'll
Hi,
fly
I'm your host,
around.
Harry Orgovan, and welcome to another episode of Our Lives, Our Future. Today, we are honored to have with us Dr. Jeffrey Seminoff. And Dr. Seminoff is the leader of the Marine Turtle Ecology and Assessment Program and director of the Stable Isotope Laboratory at the U.S. National Marine Fisheries Service, Southwest Fisheries Science Center, located in La Jolla. Welcome, Dr. Seminoff.
Thanks, Harry. It's a pleasure to be here.
Today we are sponsored by the City of Chula Vista, the South Bay Historical Society, the Chula Vista Heritage Museum, and Chula Vista Public Library. Can you tell us and start off by telling us where you were born and raised and what influences led you to the field you are currently in today?
Sure. Well, I'm one of the rare native Californians. I was born in Sacramento. However, at a young age, I moved to the Northeast. My father was a textbook salesman of all things, and I bounced around from Northern California to New York to New Jersey, and then to Illinois, and then finally to Connecticut, where I went to high school.
Oh.
But I have to say that when I was in Illinois, and I want to say this is about 1973 or 74, my first three pets I ever had were three painted turtles that I collected at a nearby pond. I named them affectionately Mo, Larry, and Curly, which dates me a little bit. I think a lot of people might not know who that is these years. But I had them all summer long and fed them tadpoles that I caught in a nearby pool. And I remember at the end of the summer when my parents forced me to release Mo, Larry, and Curly back into the pond, and I cried and just, it was a sad moment, but I think that really galvanized my love for turtles. And, fast forward 40 something years, and here I am working with sea turtles. And I just feel like the most fortunate person in the world to work with an animal and a wildlife that I love.
Can you tell us a little bit about where you went to school and what activities you did in your higher education?
Sure. As I said, I went to high school in Connecticut, and I knew that I wanted to go far away for college. I think my parents also agreed for me to go far away to college to grow up a little bit. I picked the University of Arizona in Tucson, which at the time was a school that I had some friends going to, and it looked really attractive to me. And I moved to Tucson. This is in August of 1985, and ended up doing my undergraduate degree there. And unusually, I did my master's degree at University of Arizona and then stayed at Arizona, switching colleges, and did my PhD as well, which is really unheard of for somebody that's in sort of a professional scientist to do all their degrees at the same school. For me, I had some mitigating circumstances because while I was doing my master's degree, I started an environmental education nonprofit called the Coastal Conservation Foundation. And I ended up really just wanting to stay in Tucson. I was really kind of entrenched in the community, working in local schools and things like that. And so I pitched to a professor at the university a project working with sea turtles in Baja, Mexico. And lo and behold, we were successful. I have to say, though, at the time, green turtles are also known as black turtles in Mexico, the same species we have in San Diego Bay. We're talking 1991 or so. The population was a mere fraction of the level it is today. The green turtles have recovered so tremendously, particularly in the Eastern Pacific. But at the time, back then, just what I call like major professors in my committee, they were trying to convince me to work on a different species because there were so few green turtles, and they were concerned that I wouldn't be able to get the data. But a little bit of stubbornness and not taking no for an answer, commenced to do field work in Baja. This is in 1993, and I was just really successful and surrounded myself with friends and dear colleagues that helped our project really become a success and a shining example in Mexico of sea turtle research. And mind you, this is back in the day before people really started to study sea turtles in the water. There's a little bit of work, obviously, in San Diego with Margie Stinson, but in other areas, particularly in Mexico, nobody was doing sea turtle research in the water where you actually have to go catch the animals. That was sort of unheard of. Instead, a lot of the work, and this is not just US or Mexico, but worldwide, was focused on nesting beaches, because the turtles, largely females, are crawling out of the water. So you have a very user-friendly population of animals that literally crawl to you to study. Catching turtles in Mexico or in San Diego Bay out of the water, that's a real challenge and something that we're real proud of.
And you mentioned Margie Stinson. Did you know her while she was studying the turtles here in our bay?
I had met Margie when I moved to San Diego in 2002. I just to close the loop, after I was at University of Arizona, and I knew professionally I needed to kind of spread my wings and build my portfolio a little bit. And so these days, commonly, after a colleague will get their doctorate degree or PhD, they'll often do what's called a postdoctoral fellowship, where they'll go to a university or an organization or institution for some period of time, usually a couple of years. And they're able to finish up some of their projects that they've been working on and learn some new skills from the new environment they're in, the new university. And for me, I ended up in the year 2000 moving to Gainesville, Florida, and I was at the Archie Carr Center for Sea Turtle Research, which is I'll say it's a pretty well-known spot at the University of Florida in Gainesville. And my wife and I lived there for a couple of years, and then I was offered a position at the Southwest Fishery Science Center in 2002 by my dear friend and colleague, Peter Dutton. When I moved to San Diego, quickly I learned about Margie. She's a local legend in the turtle world, and she's a delightful gal for anybody that's met her. I feel so honored to call her a friend. And though we haven't kept too much in contact over the last several years, there was a period of time where we were in close contact, and she'd come out to the field and lend us some insights about how they used to do their turtle work. I'm talking back in the 1970s. And we just were able to glean some great insights from her. And just to talk about sort of the timelessness of some of this research is that some of the turtles that Margie was working with back in the 1970s are the very same individuals that fast forward to 2002, 2003 that we were catching. So we both, in different eras, were catching the same individual turtles, which is, I think, a pretty amazing fact.
So you mentioned the Archie Carr Center for Sea Turtle Research. Now, Archie Carr, he was a founder in Green Turtle Research, wasn't he? Can
He sure was.
On him?
Yeah, he's the sort of the grandfather of sea turtle ecology, sort of our sort of shining example of somebody that's just was so knowledgeable and a really friendly person. Before I talk about Archie, I'll just say that in the turtle community, and we're a fairly tight-knit global bunch, we have an annual meeting every year where we meet and share ideas and give presentations, and let's face it, drink beer and things like that too. And everybody is just the friendliest. Sea turtle biologists, by and large, have no ego. We're so willing to collaborate, and it's a pleasure to be around my colleagues. And I've heard from others that in different sort of taxa-specific fields or other sort of science types is that it's just not the same sense of camaraderie. So I'm really proud to be part of the turtle community. And it really started from Archie Carr. And Archie was somebody that worked at the University of Florida since the early 1950s, perhaps late 1940s. He founded the first ever sea turtle research project in Costa Rica at a place called Tortuguero on the Atlantic/Caribbean coast of Costa Rica at a place called Tortuguero. And at Tortuguero, we're talking 1954. And so they started doing green turtle research there. And he's the person that came up with the idea that turtles migrate far away and then come back to the same nesting beach where they had nested earlier. And Archie and colleagues at the University of Florida started a project called Project Green Turtle. And this is in the late 1950s. And this was unheard of at the time. But what they did is they would put small flipper tags, individually coded tags on sort of the armpit of a turtle. It's a very common way that we identify individuals. And as the adults, they come to shore to nest, and they're putting their eggs in the sand, depositing eggs. They tag these animals. And then that's during the summer season. The rest of the year, Archie would travel all around Central America and the northern coast of South America, going into coastal communities, speaking to fishers, showing them an example of the flipper tag and say, have you seen one of these? And at the time, during Project Green Turtle, it had a premio and a reward of $5 a tag. So he'd bring American $5 bills with him wherever he went. And whenever a fisherman would have a tag, now comes a $5 bill out of that bill roll. And there's some places by the end of the Project Green Turtle program that he would go to communities and the fishers would have like necklaces with these flipper tags on them. Of course, it's sort of sweet and sour. They're providing information, but the only reason they got the tag is because they caught the turtle to eat. So, you know, to the turtle's demise, there sure was a lot of scientific information that was gathered from those flipper tag studies. Mm-hmm. And, you know, I could go on and on, but just safe to say that Archie was, you know, is a hero of mine. Somebody that I never got to meet. We missed each other in Tortuguero. Actually, that's the first sea turtle beach that I worked at back in 1986. But we missed each other, and he passed away in 1987. So just a great guy, great story.
So you mentioned the international community and your camaraderie.
Yep.
So you were a past president of the International Sea Turtle Society, is that correct?
That's correct. 2010, 2011, I was the president of this international society. And when we talk international at these meetings, they usually average about 120 countries that show up. So it's truly international. It's totally global. Mm-hmm. And sea turtles are essentially pan-global, except in the Arctic regions. They occur way up in the temperate areas, many spots, and of course nest on tropical and temperate beaches around the world. Yeah, and that one of the benefits I can say now, although at the time it was certainly a big lift, is that the president hosts the International Sea Turtle meeting. It's called the International Sea Turtle Symposium. Mm-hmm. And I was fortunate enough to bring that meeting to San Diego, which was the first time the meeting was ever held on the US West Coast. That was in 2011. Oh. We hosted the meeting down at the Town and Country in off the I-8 there. It's way before it got renovated, but it was a great venue, and I had a great time. Certainly stressful. We had about 1,100 people, and took a year of my life. But looking at it from this side, I'm really proud of what we did, and made new friends, and we were able to share a lot of information.
So tell me, how do you study the sea turtles we have in San Diego Bay?
Sure. Well, these turtles they're not nesting, they're foraging. They come into the bay to take advantage of the relatively warm and productive waters. Anybody that's kayaked down in the southern portion of San Diego Bay probably has seen how shallow and calm it gets. It's sort of a little just wonderful escape from the normal hustle and bustle and buzz of San Diego. Very quiet. There's a national wildlife refuge down there, and it's wonderful turtle habitat. And so the green turtles that are there have come in from nesting beaches in southern Mexico, usually at a small size, usually it's juveniles, and they set up their living in San Diego Bay and will stay for many, many years. And so what we do is we go out to physically capture the animals. We use very sort of ethical techniques. We use nets, but we get the animals out of them very quickly. Mm-hmm. The nets that we use, actually, it's a type of what's called a turtle entanglement net. Uh-huh. And they were developed ironically, by professional turtle fishermen back in the 1960s. But they're designed such that back then, the fishers would only go check their nets twice a day. They go out at 6 a.m. and 6 p.m. And I'm speaking about the turtle turtle fishery in Mexico, in Baja particularly, which is where I worked for so many years. But there'd be two people on a boat, two fishers. They go out on a panga, usually about a 25, 26 foot open skiff with an outboard motor. They check their nets at 6 a.m. and 6 p.m. And the buyers, the middlemen that would come in from places like Tijuana and drive down to Los Angeles Bay or drive down to Scammons Lagoon or Ojo de Liebre a little bit farther south, they would only buy live turtles. They wouldn't buy dead animals. And so the fishers had to figure out a way to make a net that if a turtle, let's say they leave at 6:05, that turtle gets caught at 6:10 in the morning, it's going to stay alive until 6 o'clock at night, some 11 hours later. And so they, over sort of just trial and error, I think, is they developed a net that has a very wide mesh, so it doesn't really damage the turtle. They get tangled, but not so much that they can't move their flippers. Uh-huh. There's a float line that keeps the sort of top line at the surface of the water, and a very lightly leaded bottom line that gently sort of spans out the net towards the seafloor. So when a turtle hits it, there's not really any weight or anything that's going to keep that animal from getting to the surface. And they're able to come to the surface and breathe. Now for us, we pull those animals out of the net usually within 5 minutes of the time they got entangled in the net initially. We pull them out and we bring them onto the floor of our Boston whaler. We use an outboard motor on ours as well. And we cover their faces with a damp, clean white cloth. It sort of just calms them down. That's a common technique. Covering the head's a common technique in wildlife science, used in ungulates and other animals just to calm them down so they're not seeing things or stressing out. And once we get those animals, Harry, we recognize that we're pulling this turtle out of its environment. Of course, there's some level of stress, although I'll say the stress is not like human stress, it's reptilian stress. And because of that, we want to make sure that we collect every piece of information we can off of that animal. So when an animal comes in, well, first of all, we check to see if it has any flipper tags, which is that external tag from the Project Green turtle that Archie Card basically invented back in the day. We look to see if it has a flipper tag, and we also tag turtles with this internal tag called a passive integrated transponder. It's like a little grain of rice. It gets injected into the top of the flipper with a sort of a needle. Folks that have pets that have been chipped, it's basically that same thing. It's called a pit tag, the chip that you hear of in dogs or horses or things like that. And so we'll scan that animal with a special scanner to see if it has an existing chip in it. Sometimes the flipper tag's fallen out and the chip's still in it. That's the benefit of the chip. And if not, we'll put new tags on that animal. We collect blood from that turtle, right from what's called the dorsal cervical sinus in the back of the neck. We're all gloved up, total hygienic situation. And then we will, with that blood, we can get a lot of different information. First of all, we can find out its genetics, sort of its DNA fingerprint. From that, we can kind of figure out its nesting beach origin, like where it came from, what stock it came from. We also analyze blood to look at hormones, so things like testosterone. Small turtles, males and females, are indistinguishable. The only way you can tell the difference is through external inspection, internal inspection of the gonads, or of looking at testosterone levels in their blood through a calibrated approach. And so we'll look at testosterone in blood. We'll also look at stable isotope values in blood, and that can give us some idea of the diet of that animal, that very individual. Is it a total herbivore? Does it eat plants and a little bit of meat? Does it eat nothing but meat? It's amazing with something called stable isotope analysis. With just a minute amount of blood, you can tell those. You can answer those questions. And then something more recently that we're doing with hydrogen, with something called stable hydrogen, is we recognize that hydrogen levels in the blood vary depending on the reproductive status of that animal. So we use testosterone to tell if it's a male or female, and we will use hydrogen isotopes to tell if it's in reproductive status or not. So we get a lot of information from blood. And then we also, of course, will work up a whole blood panel, much like a veterinarian would for your dog, to look at percent cell volume and various nutrient levels and things like that. So in addition to the blood, we take photos because we like to photograph every individual. So we have sort of just a photo documentation of those animals. Oftentimes, if a turtle loses a flipper tag or it wasn't pit tagged and you've caught it multiple times, you can tell individuals based on some external scars they might have. On occasion, we'll have an animal that's missing a front flipper. They can still swim quite well with just one front flipper, trust me. But photographs come in really handy. And then the other component that we do, which is really focused on understanding kind of where animals move and how long they stay in the bay, is we employ something called satellite telemetry. And with satellite telemetry, we apply a tag on the top of the animal that has a small antenna, and that transmitter will transmit information about the location of that turtle as it swims around the bay, or even as it leaves the bay to go down to nest in Mexico. And so that's sort of the whole spectrum of the things we do, from capture to blood analysis to telemetry. I forgot to mention weighing and measuring and things like that, just to get some idea of the size.
Sure.
And with that, we sort of have this holistic approach with how we're studying green turtles in San Diego Bay. And I should point out that, we talked about Margie earlier. This is really on the shoulders of Margie. She was the one that instigated this. And then the next person in line, which really brought it to the next level, is Dr. Peter Dutton, who's, again, as I mentioned, a close friend and colleague at our Southwest Fishery Science Center. And
Mhm.
Peter, even in the late 80s with his wife Donna, started to go out and observe these turtles and started catching those animals. And so it's really, I kind of can call this the San Diego Bay stuff like a Dutton legacy project, because he is the person that really made this happen. And I'm just kind of carrying the torch that he started like way before I moved to San Diego.
So what can you tell me about natal homing and how that works? And what age do the turtles in our bay, who I guess are mostly the Eastern Pacific green sea turtle, what age do they reach maturity in our bay?
Great. Yeah. Well, for the longest time, we thought that green turtles reached maturity in some 30 to 35 years. This is back in the 70s and 80s. But more recently, what we've discovered is that they mature at much younger ages than that, perhaps a response to the low population size from those decades of harvest. But we expect turtles, and it varies depending on sort of nutritional status of the animals, but anywhere from 20 to 25 years old, that animal will reach maturity. And so what happens though in the general life cycle of the sea turtle, if we take it from an egg and move forward, is that turtle, let's take green turtles, and the primary nesting beach for green turtles in the Eastern Pacific is in the state of Michoacan in southern Mexico at a beach called Colola Beach, a little bit of a tongue twister there. And at Colola Beach, they've been doing work since the late 1970s. But a green turtle, locally there, they call them black turtles. They'll crawl to shore. She'll dig an egg chamber. It'll be deep, sort of in a body pit in the sand. This whole nesting event can take upwards of 3 hours sometimes. The green turtles will lay upwards of about 100 eggs, maybe a little bit less. They're a little bit bigger than a ping pong ball, a little bit smaller than a racquetball. They're leathery to the touch, so you can actually touch it and push on it, and it will dimple in. Actually, and as those eggs develop, they take an ambient water and they grow in size and weight throughout that incubation period. The incubation period for an egg is about 55 days, give or take. And during that period, what's really interesting is that as that embryo is growing, something that happens with all sea turtles and most turtles, but not all turtles, is called temperature sex determination. And what I mean by that is the temperature of the developing embryo, and this is about in the middle third of the incubation cycle, about in that middle third, if there's warmer temperatures above about 29.5 degrees Celsius, the embryo will grow up to be a female. If it's cooler than 29.5, that animal will grow up to be a male. And so what happens with this temperature sex determination is over the course of the nesting season, where it'll span kind of cooler periods of time and warmer periods of time, you'll have a preponderance of one sex produced during one portion of the nesting season and the preponderance of another sex produced at the other portion of the nesting season.
Yeah.
In addition, even within the egg clutch itself, the eggs that are closer to the middle, that 100 eggs is going to be about 8 or 9 pounds. It's going to be about the size of a bowling ball. Those eggs in the center, there's been so much embryonic heating from their development, as though those eggs are warmer than the outer eggs. Oh. And sometimes when nests start incubating and the ambient environment is right around that pivotal temperature, around 29.5, and that pivotal temperature may shift back or forth depending on the population of species. The eggs in the middle will turn out female, and the eggs on the outside will turn out male. So at the end of the day, we expect this about a 1-to-1 sex ratio. And then at 54, 55 days, those animals kind of, they pip, they crawl out of their eggs, and they all are timed to do it at the same time, and they wait. They'll crawl. And if you ever had a bucket of sand and you put some rocks in it down at the bottom of the bucket and you kind of shake the bucket, the rocks always work themselves to the surface. And there's a similar dynamic with sea turtle hatchlings. You have 100 hatchlings that are kind of milling about down, it's usually about 2.5 to 3 feet deep, and they slowly work themselves to the surface. And what they do is, as they get towards the surface, once they sense a temperature heating, which usually occurs in the top 2 or 3 centimeters, which is usually because of solar heating during the middle of the day, they stop and they'll go into a stasis until the sun goes down, until that temperature cools off, and that's sort of like that barrier cools off. And then they sort of explode out of the sand and all scurry to the water. Now, another thing that's happening during that incubation period is they're imprinting on the local location, and they have something called magnetite in their brain. They basically create a natural compass. And with that natural compass, they kind of have their GPS spot of where they came from. And so once they kind of emerge and burst out of the sand and scurry to the water, and typically hatchlings, when they're going to the water, you talk about navigation, is that what they're looking for is a slightly brighter light. And so usually in the horizon behind them where there is jungle and forest, it's going to be a lot darker than over the ocean that's going to have some reflection of stars and things like that. And even to the human eye, sometimes we can't even sense those minute differences, but turtles can. And so they go, it's what's called sort of like positively phototropic, and they go towards that light. Well, one of the problems with hatchings, of course, and this particularly has been an issue in southeastern United States, although they fixed it with responsible lighting techniques, is that artificial lighting makes all those turtles crawl inland. And so a lot of times you'll see a bunch of turtle hatchling chips, dried up dead turtles in the morning, below these streetlights that were near the beach. Fortunately, many places worldwide realize that it's an easy fix. So there's a lot of turtle-friendly lighting out there is what we call it. But so once they hit the water, and meanwhile, Harry, they're already dealing with the onslaught of predators because they've got ghost crabs, they've got seabirds coming at them, they've got raccoons, they've got feral dogs, everything when they're on the beach. And then once they get into the water, they have more seabirds and they have large fish and stuff like that. And so generally speaking, very few of those hatchlings that actually emerged from the nest survive to get offshore. But the lucky ones that do, and we generally use the rule of thumb as from 100 hatchlings, one of them is going to survive to adulthood, more or less. There's probably less with the human pressures and fisheries impacts and things these days, but historically we've generally said about 1 out of 100 to maturity. And then so once they hit the water, the hatchlings will begin what we call an oceanic phase, where they'll stay offshore for some 2 or 3 years. Let's go back to Archie Carr for a second, because Archie Carr phrased a term called the lost years. And what do I mean by the lost years? Well, that is that as scientists and as just humans, we would see those small hatchlings crawl into the ocean, and we'd never see any turtles again until they were about dinner platter size. Just where are those small turtles? And Archie Carr termed this term lost years. And in between his exploits with the Project Green Turtle, he also mounted a number of efforts offshore to look at the flotsam and driftsam rafts of debris and sargassum and things like that offshore. And lo and behold, he found that sort of missing size class living in the high seas, far from the shore. And so those animals stay in that lost years period. It's a lot of the research we do at our Southwest Center lab, looking at how long they spend offshore. And we think it's between 3 and 7 years, depending on the individual. And then there's a little bit of chance there. Sometimes animals will just randomly get swept inshore, or some animals get swept offshore, so it prolongs or it minimizes that lost years period. But something clicks in their brain at, I want to say, again, like 3 to 7 years old, probably closer to 7, where they want to change their habitat. They want to live in a different environment. And what we call is animals that transfer through these different life stages. Those are what are called ontogenic life stages. And there's this thing called an ontogenic habitat shift, which means that as animals mature, not just sea turtles, as they mature and become hatchlings to adolescents to young adults to adults, their preferences for where they live change. Kind of like humans a little bit, I think. And so what happens is those turtles at that 5-ish years old, they want to then undertake a coastal existence. And that's when we start seeing animals settle back into coastal habitats like San Diego Bay, like up in La Jolla, Seal Beach, San Gabriel River. Throughout Southern California, we have a lot of green turtles in coastal waters. And they then kind of live in those areas for a long period of time. And much like animals have that nest site fidelity, or that philopatry, where they'll return to the same nest that they came from, once they settle in on a habitat like San Diego Bay, they'll stay there sometimes for the rest of their lives. And to give some perfect examples, we have a number of green turtles that we have caught over the years for more than 20 years. Take Margie Stinson's wrinkle-butted turtle. Maybe we'll talk about that a little bit, but wrinkle-butt was the largest green turtle in the Eastern Pacific ever known. First, I think, seen in 1976 by Margie, and then we last captured her in our NOAA research in 2007. At the time that we captured her last, she weighed 540 pounds. 540 pounds. That's a big turtle, because they average, the big ones will usually be like 300 pounds. And so, but we've had plenty others where, not only have they stayed there for that long period of time, 20 years, because we capture them periodically in the same habitat, but we've put transmitters on those animals, and we have satellite tracks of females and a male, 3 females and 1 male. They actually left San Diego Bay, went down to southern Mexico to nest, spent several months down there, and then came right back up into San Diego Bay following a very similar path, and then they just set up camp again. So San Diego Bay offers everything that a green turtle needs. And so that's why we have this thriving population there. And when you open this, Harry, talking about sort of that nest site fidelity, the navigation components,
Yeah.
is that amazingly green turtles have it on kind of both ends. They know where to go to nest. So when they leave San Diego Bay as adults, they go back to the exact same beach they came from, sometimes within the same 100 meters of beach. And they know where they foraged. And then after nesting, they go back to where they foraged. And so it's just an amazing cycle that these animals, are able to kind of go back and forth in this big loop throughout their lives.
When you find a turtle perhaps that has passed away on the shore and you can still study them, what are the components you utilize to study a turtle, and what things are you looking for when you find a turtle that has passed away?
Yeah, so when an animal strands on the shore, or we find a dead animal on the shore, typically we refer to that as a stranding. It could be a dead stranding, like a dead dried up turtle on the shore or recently killed, or it could be a live stranding. Sometimes we have animals that are just decimated and sick for some reason. And so in Southern California, we have a pretty solid network of stranding response. And that network includes places like the Aquarium of the Pacific up in Long Beach, California, and our West Coast regional office in Long Beach as well. Includes a number of smaller organizations up the coast, all the way up to Point Conception, Marine Mammal Center, for example. Here in San Diego, we work very closely with SeaWorld. SeaWorld is a big partner of ours. They've been a sort of a wonderful helper of sea turtles in Southern California. The way it works in terms of the stranding arrangement is if an animal ever live strands, like it's still alive, SeaWorld responds. Or if it's up north, Aquarium of the Pacific responds. If it's a dead turtle, then my team responds. And I have to say that over the last year, during sort of this COVID period, we've had very few strandings in Southern California. And I want to say I can count on one hand how many dead turtles we've had along our coast, and we have pretty good coverage. And perhaps that's a result of less traffic on the water and things like that. I don't know, but in years past, we would average anywhere from 10 to 20 dead turtles a year on our coast, which is very little when you compare to the U.S. East Coast, where NOAA and a variety of state and local agencies work together to respond to strandings, where they have 2,000 to 3,000 turtles a year. So we have a very sort of utopic situation in California that we just have so little sea turtle mortality, which is quite nice. But when we do respond to an animal, if it's live, it goes into the husbandry tanks at SeaWorld or Aquarium of the Pacific, and they get them back to health. We're actually, in a couple of weeks, SeaWorld's going to release a loggerhead turtle that was live stranded some 3 or 4 months ago that was in horrible shape, and it's ready to be released. And just today, I was at our lab with special permission because of COVID to pick up a satellite transfer of them and to provide to SeaWorld to use. So we collaborate quite a lot on some of these things. For us, with dead turtles, we will, first of all, if we encounter it on the beach, we'll take that scanner to see if there's an internal pit tag on it. We'll look externally to see if it has that flipper tag, the Archie Carr type flipper tag. If they have those tags, then we know exactly which individual it is. And commonly, when turtles used to strand in San Diego Bay, and in the Bay, they largely stranded because of boat strikes. A lot of times, unfortunately, it would be turtles that we've been studying for a long period of time. But so we've checked for flipper tags and pit tags to see if those animals are, individuals that we know. We'll take a skin sample off of that animal right there. Usually we try and do some processing right on site just to get it done. There's a big data sheet we fill out, location, body condition, like how decomposed is it, things like that. And then we will take that animal, put it in the back of our truck, and we bring it back to our NOAA facility. If we have the personnel, we'll do what's called a necropsy right away. That's sort of like an autopsy, but for wildlife. When we do encounter dead turtles, as I said, we're collecting all the different sorts of information we can on it right on the spot. Then we bring it back to the lab. If we have the human resources at the time, we'll do what's called a necropsy. It's sort of like an autopsy, but for wildlife. If there's nobody there, we'll put that animal into the freezer until we can do the necropsy. And that necropsy is very important because oftentimes we can determine the cause of death. And we work very closely with a number of veterinarians. Dr. Heather Harris is one of our primary vets that we work with closely. Eric Otchen and his team at SeaWorld are people we work very closely with. And we can just tell if it was a boat strike or if that turtle died from some other reason and then it got struck by a boat as like a secondary injury after it was already dead. Those are the sorts of things you can tell with a necropsy. We'll look at its gut tract to figure out what the animal has been eating. A lot of times we can tell if there's been some sort of intestinal blockage because of eating plastics. Plastic ingestion is a big problem with sea turtles. And then the other final thing that we'll do is that we will collect a bone out of the shoulder. And that bone's called a humerus bone. And the humerus bone in sea turtles deposits annular growth rings. And it's much like trees, how trees put down rings every year, and you can chop down a tree and you can count the rings and figure out how many years that tree's been alive. Mm-hmm. Well, lo and behold, with sea turtles in that humerus, they also lay down these annular growth rings. And whereas with trees, they use what's called dendrochronology. With sea turtles, we use something called skeletochronology, where we're looking at the chronology in the skeleton. And with that sort of information, and if we look at the isotope values in those various layers, first of all, we can count the layers and find out how old that turtle is. Uh-huh. But secondly, we can figure out how long that turtle's been in our local habitat, or how long it's been living offshore in those lost years periods. Uh-huh. So one dead animal can yield just a wealth of information scientifically for what we're trying to get done. And probably more importantly, it yields information about what the causes of death are, so that management can figure out things like where we need to slow boats down or where we need to stop fishing or where there's huge problems with marine pollution. So it's amazing what an animal, even as it's a dead animal, can help us out with sea turtle conservation.
Yeah, I've heard in San Diego Bay, one of the main pollutants would be PCBs. Mm-hmm. And
yeah, so that's a little bit out of my realm of expertise. I certainly know that the Unified Port of San Diego has been trying their best to clean up the bay, and I think they've done a great job in many cases. Mm-hmm. They've done a lot of seagrass restoration throughout the southern portion of the bay, which is great. I think there's a lot of local regulations that try and minimize inflow into the bay after rains and things like that. Mm-hmm. But I think like any industrial waterways, certainly there's heavy metals and organic pollutants, PCBs, DDTs, copper, lead, mercury, things like that. And we've had graduate students throughout the years that have done a number of studies. And I failed to mention with our blood, we also look in contaminants. So add that to the information the blood can yield. But what we found is that these sea turtles will have extremely heavy contaminant loads on occasion. But for some reason, perhaps to the physiology of sea turtles, and it just sort of speaks to their resilience, is that they rarely show any sort of acute health effects from that contamination. Turtles thrive in contaminated water. I'm not saying San Diego Bay is contaminated, but there are plenty of contaminated spots, and sea turtles just thrive in those areas. For how that works, I don't know. But one of the things that's worth mentioning as we're talking about green turtles is an area where we know that disease is a big problem, or a type of disease, is something called Fibropapillomatosis. And it's a herpes virus type affliction. So we have graduate students that have on a number of occasions studied the contaminant loads in green turtles in San Diego Bay and up in Seal Beach National Wildlife Refuge, as well as the San Gabriel River. And what we find is that on occasion, animals do have heavy pesticide or organic pollutant loads in their blood. It's another item that we study in blood I failed to mention earlier. But we found that despite heavy loads, they don't really reflect any sort of acute health effects. Turtles tend to be really healthy. They are fat and happy and eat well, even with those high contaminant loads. Now, there might be a threshold at which it leads to their demise, but we really haven't found any cause and effect of pollution with sea turtle mortality or poor health. But one of the areas that there's a big outstanding question with the cause of this, it's an affliction called Fibrocapillomatosis. It's this really unsightly coliform, like cauliflower type tumors that grow on turtles. It's a herpes virus. Mm-hmm. It occurs in great prevalence in places like Hawaii, which seemingly are pristine, as well as Florida. and many other places worldwide. We really don't have any sort of direct cause and effect with the fibropapillomas, but we do believe that it probably has to do with some sort of coastal runoff of pesticides, perhaps some sort of immunosuppression that happens because of pesticides. And then there's sort of this third-party virus that takes advantage of turtles that are immunocompromised. And that's what kind of gets the turtles. There's been some evidence that fibrocapillomatosis also is physically transmitted, so turtles touch each other and they give it to each other. But at the same time, we've also found that some individuals regress with their tumors and the tumors go away. So still a lot of important veterinary science to be done with fibrocapillomatosis. Fortunately, we don't have those big cauliflower-like tumors on our turtles in San Diego Bay. Although Peter Dutton and his team in years before I came to San Diego Bay did find a turtle that had tumors that were of fibropapillomas type, but not the same strain as what's found in Florida and Hawaii. So we hope we never do see it in California.
So in the south back part of San Diego Bay for the longest time, from early 1960s through September of 2013, we had the South Bay Power Plant. And the outflow from that plant gave us warm water, perhaps 80 degrees Fahrenheit and above. And the turtles used to conjugate more in that back part of the bay where the outflow was and hang out there mostly. So now that the power plant has been imploded and removed, what are the effects of that in relation to the turtles?
Well, fortunately, we have answers to those questions, Harry, because we did a lot of that telemetry and tracking those turtles' movements in South Bay, both pre and post power plant destruction. And what we found, I want to say we tracked about 40 or 50 individuals, thereabouts. What we found is that once the power plant closed and essentially that hot water spigot was turned off, is that turtles started to venture much more into the central portion of the bay, north of the Sweetwater Channel, for example, up towards the Coronado Bridge and over along Silver Strand area. And so also commensurate with that power plant closure is we did see an uptick in boat strikes. And that's probably because turtles no longer were tied to that warm water in the southern portion of the bay and they're swimming around. And there's a lot more boats as you go farther north in the bay. But I would say that, green turtles, and this speaks back to like, how did they get here to begin with? And there are some theories that green turtles, this population was seeded, if you will, by escapees from the old days of the turtle fishery when they used to boat up boatloads of green turtles from Baja, Mexico to feed locally and also ship up to San Francisco and other inland areas. There's also been suggestions that that warm water is what brought them here and that it's the only reason that they're here. But actually what we believe now, and we have data to suggest this from our skeletal chronology work, is that these turtles, it's a natural population. They were here long before the power plant. They were here long before there was any humans fishing turtles in Baja. It's a dynamic population. We know that the Pacific lagoons along the coast of the Baja Peninsula, they're even a little bit cooler than San Diego Bay, and green turtles thrive in those lagoons. At the same time, if we go up to places like off the coast of La Jolla, in that region where it's extremely cold water, relatively speaking, the turtles thrive there. So it wasn't really the warm water that kept them there or attracted them there necessarily, but they sure took advantage of it. And back in the day, pre-2011, 2012, green turtles could maintain activity throughout the year. Even in the wintertime, they had warm water so they could eat year round. They grew faster. Once the power plant closed, the return back to sort of ambient conditions, and in the wintertime, turtles tend to hunker down. And whereas in years ago, we used to catch turtles only during the wintertime because that's when it's kind of the easiest to catch them. Now we only do it during the summertime because during the winter, they're kind of asleep on the bottom and they're just not moving very much. And so the habitat has certainly changed incredibly, but it's been sort of a natural experiment because here we have this artificial hot water situation back 10 years or so ago, and then immediately was turned off. So we could see the response of the turtles and we could see the response of the habitat. Anybody that spent a lot of time back there knows that like the seagrass habitats have really started to come back. A lot of that silt over at the old channels of the South Bay Power Plant are starting to fill in. You can't quite get your boats in there the way you used to. But I think that's a good thing, because I think that reflects a return to a natural state. I think some of the exciting work that we hope to do once COVID relaxes, and I have to say, unfortunately, Harry, we haven't been in the bay since 2019. We missed all of 2020. And here we were getting ready to go back into the field this month, but this Delta strain of the virus has made our lab shut down again. So we had the rug pulled out from under us. It's going to be going on 2 full years that we haven't been out there.
Oh.
But one of the things that we're very excited to do is to use a new kind of technology that's called turtle-borne video, where we actually have a system that we'll use to put on the back of the turtle that stays on for some period of time and automatically pops off. And we are able to track the movements of those animals, but more importantly, see how they live underwater and what choices they make. Do they really depend on seagrass the way we think they do? Are they eating small snails on purpose, or is that accidental consumption? You know, questions like that we can start to address when we actually have sort of a turtle's eye view of what those animals are doing. As soon as we get back in the field, we have our new research tool, or toy, maybe some would say, that we're going to use to learn just even more things about the lives of green turtles in San Diego Bay.
Well, Dr. Seminoff, I really appreciate you being with us today. You're an incredible person for all the work you've done, and you're proceeding in that venture. And I just want to say this is one of the fields I really like, and you're an expert in it. And we really appreciate everything you and your team are doing.
All right, I sure appreciate it. You know, thank you. Thanks to the Living Coast Discovery Center. I have such respect for what the organization does. Really important for wildlife rehabilitation and husbandry. And I think you do a great job of bringing green turtles to the public, to people that have never seen them before, even kids from that same region that don't have any beach to visit, but they get to the Living Coast Discovery Center to see those animals in action. So thanks again to you and everybody else at Living Coast. I really appreciate it.
Oh, appreciate that. Thank you, Dr. Seminoff, and thank you everybody for joining us on Our Lives, Our Future. Bye-bye.
Bye-bye.
Pa is rich,
Ma is rich.
so now what do we care?
I love to be beside your side.
Beside the sea, beside the seaside, by the beautiful sea.